Hello, welcome to lesson 7.3. Today we'll be talking about bacterial feeding nematodes and their particular role in nutrient cycling. So we'll look at the importance of nematodes feeding on bacteria and how that affects plant growth as well as nutrient availability in soils. Okay, let's jump into lesson 7.3. So let's look at bacterial feeding nematodes. About 40% of total nematode numbers in soils are actually bacterial feeding nematodes. So they comprise the majority of soil nematode community. When microbial activity is greater, they can actually comprise 90 to 99% of all the total nematodes in soil. They regulate bacterial abundance and community composition, and that affects carbon mineralization rates. They are absolutely essential in soil ecosystems as they alter energy flow and nutrient cycling. They are, in fact, besides protozoa, or bacterial-feeding protists, the only organisms that can actually unlock microbial, in this case bacterial, nutrients and release those nutrients. So without nematodes, if there are also no protists, there is no way of releasing nutrients from bacteria. And remember, bacteria get those nutrients directly from the soil matrix. And without that consumption of bacteria and the transformation of those nutrients from within the bacteria, there is no nutrient flow further on up the food web. And this is just vital to understand that without the predators or grazers of bacteria, there is no release of the very essential bacterial nutrients that bacteria pick up directly from the minerals. This is where those minerals then enter the nutrient cycle and then they feed every other organism that exists within the food web beyond the nematodes. This is where we get minerals from. So absolutely key organisms to have in the soil. When there is increasing grazing pressure by nematodes on bacteria, bacterial biomass and diversity actually increases. And that happens up to a point, of course, where there's a regulation of that grazing pressure and it's at its optimum. That's when bacterial biomass and diversity is at its highest. They can consume around 5,000 bacteria per minute, but 60% of consumed bacteria are actually defecated alive, along with growth factors that could include limiting nutrients and hormones that the bacteria actually rely on, and therefore that causes a bacterial population increase. They increase bacterial transport, both internal and external, both in their guts and on their cuticle, on the skin. So they basically function as bacterial dispersal mechanism in soil ecosystems. They also create physical and behavioral bacterial refuges, which allows colonization of new niches. So what this means is that by nematodes existing in soil, they actually change the physical properties of soil. They increase soil aggregation and create pore space, for example. And so that allows bacteria to colonize new niches. And at the same time, it changes how the bacteria behave. And that's because these little aggregates that are formed means that some bacteria can hide in them. All kinds of potentials are possible by these bigger organisms moving throughout the soil and therefore creating habitat for their bacterial prey and stimulating that prey. Soil disturbance always increases bacterial populations. Once again, it creates a new niche for the bacteria to colonize. And this is a beautiful image of two nematodes here. And a really good reference paper for this, which we'll use in the next slide, is by Ingham and others, looking at the relationship between bacteria, fungi, and nematodes and how that affects nutrient cycling and plant growth. So let's look at how presence and activity of bacterial feeding nematodes functions in nutrient cycling in soils. We'll use an example from this research paper by Ingham and others, because it showed a really nice picture of the importance of bacterial feeding nematodes. So bacterial feeders consume bacteria and protozoa depending on how large the mouthparts are of the various species of bacterial feeding nematodes. Since the bacteria and the protozoa have lots more nutrient concentration than the nematodes themselves, the nematodes then have to release the excess inorganic nutrients, and this accelerates nutrient cycling. By doing so, they increase plant growth and plant nitrogen uptake and mineralize nitrogen and phosphorus, and this increases bacterial populations. When bacteria and nematodes are present in unison, this increases bacterial densities and plant growth and initial nitrogen uptake by the plant, versus when no nematodes are present. So if only bacteria are present, none of this actually occurs. This is because nematodes increase bacterial nitrogen mineralization as they consume bacteria, and they deposit the excess nitrogen in the form of ammonium into the soil. And this stimulates initial root growth and also root branching, which is really important for nutrient access by the plant. So they essentially create a more robust root system for the plant and therefore set the plant up for healthy growth. The mineralized nitrogen immediately becomes immobilized by the bacteria and fungi and other organisms that live in the soil that take up this mineralized nitrogen, or this nitrogen can also be taken up by the plant. So there's a little bit of a competition going on when that nitrogen enters the soil as the bacterial grazers defecate. So the bacteria and fungi, the microflora, actually limit plant nutrients unless there's predation going on. And you can imagine this in a way that while all the bacteria stay alive in the soil, they are holding on to those nutrients in their bodies. So there's got to be some sort of an unlocking mechanism to access these nutrients from the bacteria and fungi, and that is when predation becomes really important at critical plant development. An example of that from another study is that when nematodes were eliminated from the soil matrix that was studied, there was less nutrient mineralization and wheat nutrient uptake throughout the whole life cycle of the plant was reduced. There was also a reduction of phosphate uptake at seed formation and maturity, therefore significantly affecting the growth and health of the wheat crop. So this just shows how important it is to have bacterial feeding nematodes in the soil ecosystem. This beautiful image shows the intricately adorned mouth. So really these are the lips of a bacterial feeding nematode. Up to 20% of ingested bacterial nitrogen and 75% of ingested phosphorus becomes available for plant uptake when bacterial feeding nematodes release these from bacteria. And the scenario is that the greater nematode carbon to nitrogen ratio, as compared with the lower bacterial carbon to nitrogen ratio in bacteria, leads to the release of excess soil nitrogen. This is because the nematode has more carbon than nitrogen, and by eating bacteria that have less carbon than nitrogen or more nitrogen than carbon, by default, there is too much nitrogen for that nematode now in its body. It has to deal with this because it doesn't require as much nitrogen as the bacteria that the nematode feeds on. So that nitrogen has to be released because it's in excess. So basically the nematode defecates, poos out the nitrogen into the soil. The greater the organic matter carbon to nitrogen ratio, so the more carbon there is in the organic matter that the microbes feed on, the greater that carbon to nitrogen ratio is, it generally means that there is decreased net mineralization or lower concentrations of nitrogen in the soil as a result. This applies to, for example, if we have a lot of woody debris in the soil that has more carbon than nitrogen, that will initially mean that there's less nitrogen being mineralized. This is why it's important to have all kinds of organic matter added to the soil with various carbon to nitrogen ratios. So diversity of feed is important because that will then generate diversity of bacteria and fungi, and then that will also feed the various nematode feeding groups. So there is a lower expected nematode mineralization by consumption of bacteria at greater carbon to nitrogen ratio versus when those ratios are lower. So basically it's the same concept explained in a different way. So greater carbon to nitrogen ratio leads to greater carbon source for the bacterial community to feed on. However, it is limited by nitrogen. So that nitrogen has to come from somewhere, and if the nitrogen is being released by nematodes, even if that carbon to nitrogen ratio exists in some of the food sources that are added to the soil, that nitrogen will not be limiting as long as there are nematodes present and doing the nutrient cycling from the bacteria that they feed on. So this just basically explains the extreme importance of food source biodiversity in the soil, the organic matter variety that is added, and how it relates to the microbial / predator and grazer interaction, and therefore how that links to nutrient cycling. So diverse microbial communities and sources of organic matter provide optimal plant growth, and both are very necessary. And of course, when the sources of organic matter are varied and diverse, then that will translate into diverse microbial community, and this always translates into optimal plant growth. This is an image of another beautiful bacterial feeding nematode called Acrobelis, and once again it has some intricate lips that adorn its bacterial feeding mouthparts, no doubt to capture the specific bacterial food that it prefers. So let's look at how bacterial feeders influence carbon cycling. Greater bacterivore abundance increases grazing pressure, and that increases microbial-derived soil organic carbon turnover. So bacterivores increase easily available carbon compounds, rhizodeposition. So this means that as the bacterivores feed on bacteria around the rhizosphere of the plant, they actually stimulate the release of carbon compounds as they process the bacteria around the root zone, and also stimulate the bacterial activities to decompose carbon, and therefore by feeding on those bacteria, they further break down the carbon compounds, ensuring that these carbon compounds are available in the soil rhizosphere interface. Through predation, they increase bacterial abundance, and this causes carbon sequestration because the bacteria actually take up the carbon into their bodies, and so do fungi and other organisms as well. This is incorporated back into the life forms. So this sequesters carbon, and it causes carbon accumulation and storage rather than release back into the atmosphere. So this is really important, that predation basically holds the carbon bound in the soil matrix. Also, the bacterial feeders themselves require lots of carbon. They have very carbon-rich cuticles, so this further increases carbon accumulation in the soil as it is held in the biological life forms. All the nutrient cycling that occurs via nematode grazing on bacteria is especially important at low nutrient availability in soils, and this is often encountered in nutrient-limited natural soils. So having bacterial grazers in all soil ecosystems, but particularly those that are nutrient poor, means that the nutrients are retained within the ecosystems. It means that they are not lost, they are not leached out, but actually scavenged continually and being maintained in the loop for the plant, and to maintain the microbial populations through bacterial grazers doing their job and cycling those nutrients from the bacteria. This beautiful image on the right is of C. elegans, the nematode that is used for all kinds of medical studies, and it's an NMR spectroscopy image by Queensland University where they were looking at the nervous system of the nematode and studying it to solve some of the nervous system disorders and damage in humans, and how that can be repaired looking at the C. elegans model. Just a beautiful image. Another important way in which bacterial grazers actually enhance plant growth is through their interactions with particular bacterial groups. You would remember that plant growth promoting rhizobacteria help plants to function. And one of these groups of bacteria is one that produces auxins. Auxin is a plant growth promoting hormone, and some of these bacteria actually produce these hormones to help plants grow. And this particular study looked at the effects of the interaction of auxin-producing bacteria and bacterial-feeding nematodes on the regulation of peanut growth. The particular auxin that they studied was called indolacetic acid, which is this acronym here, and the bacteria that produces it is called Bacillus megaterium. And there are two bacterial feeders that were used in this study, the two different nematodes. And they were assessing how the presence of these players affects peanut growth. So when bacteria and bacterial feeders were present, there was an increase in soil nitrogen and phosphorus availability to plants, and therefore increased plant nutrient concentrations. So this, of course, led to increase in plant growth, as well as an increase in root branching length and thickness. It also stimulated arbuscular mycorrhizal fungal growth, which is wonderful because then the fungi themselves also access minerals and nutrients for the plant, particularly phosphorus. When bacteria and nematodes were present, their effect was much greater than when just nematodes were present or when there were just bacteria present without nematodes. So both act synergistically because the nutrient cycling, the nutrient release is happening through the grazing mechanism. Bacterial grazing increased nitrogen and microbial biomass and activity, as well as microbial community and diversity in that community, and soil indolacetic acid, so the hormone produced by the Bacillus megaterium bacteria. This is a gorgeous image showing one of the species studied, Cephalobus species. So this study showed also that the effect on peanut growth was nematode species dependent. So up until 42 days of peanut growth, the results were pretty much the same. But after 42 days, one of the nematode species, Mesorhabditis species, showed greater nutrient turnover and plant growth promoting effects versus the Cephalobus species. And this is most likely due to the different feeding habits of these two different nematodes, where Mesorhabditis is non-selective at high bacterial rates versus Cephalobus species, which is actually a selective consumer. So how these organisms feed and their growth rates in terms of how fast they can actually grow and reproduce and which bacterial species they feed on affects plant growth. This is once again showing us that we need an abundance and diversity of various species in soils in order for plants to receive the greatest benefits from those interactions. So bacteria, nematodes, and incubation time interactions significantly influence plant growth, plant and soil nutrient concentrations, and soil indolacetic acid concentrations. Bacterial-feeding nematodes are known to stimulate indigenous plant growth promoting rhizobacteria activity and therefore increase soil auxin concentrations, as well as many other benefits that plant growth promoting rhizobacteria actually provide to plants. So this image is that of the Mesorhabditis species. We have come to the end of lesson 7.3, where we've introduced the importance of bacterial feeding nematodes in nutrient cycling. And in our next lesson, we'll continue talking about bacterial feeding nematodes, but look at specifically the nematode-bacteria relationships or interactions. And we'll also look at how soil habitat affects these relationships. Okay, you'll be hearing from me again in lesson 7.4. Bye for now.